Okay, here goes.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured.
Okay, keep in mind, insulin goes outside of the cell, so it's going to go through the secretory pathway. So she's probably going to use that pathway to talk about this.
The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
Okay, what enzyme was that? She didn't tell you.
RNA polymerase. RNA
polymerase. And you could see in that video how there wasn't very much of the DNA that gets separated, and it's not very much of the RNA that stays hybridized. The DNA goes back together pretty easily, and the RNA kind of drifts off.
The information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected.
Okay, what are those enzymes called?
Is it the tRNA?
Nope. We're not to the ribosome yet. What causes splicing? The spliceosome. Good, 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 good.
Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches.
Notice it didn't bind at the very beginning 3 nucleotides. It had to read along till it found that start codon. What's that start codon again? AUG, that's right. Now, don't worry, on the test, you will be given a genetic code. You never have to memorize that, but it's always good to know that start codon. Just makes it easier.
A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the 2 amino acids.
What's that bond called? Close. That's what the whole growing protein chain is called. What's the bond? Yeah, just a peptide bond, that's right.
The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids.
What's the minimum number of tRNAs you have to have? 20, that's right. And probably that is probably the case because of wobble. You don't have to have multiple ones, like if serine has 4 different tRNAs that fit any of those 3 tRNAs will be charged by the same enzyme, likely.
In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Did that help a little bit? Seeing it in kind of real time? Yes? Okay, good. Excellent. All right, so we have a little bit more to talk about in chapter 14. So what I told you before, remember that this process, the whole idea of the central dogma is universal, which means you can take the gene from a firefly that codes for the firefly's glowy butt and put it into any other organism and express it, as long as you have the right. Now think about this in terms of all of the control we have over it. You have to have the right promoter or a promoter that's going to make sure it's expressed, and then you have to have the right transcription factors to make it expressed. One of the things you can do easily if you want to express this gene somewhere else is you find a promoter in this organism, in the plant, and put this gene under the control of that promoter. So now the plant is not going to know what gene it's making. It just knows that it's supposed to bind to that promoter and make that particular protein. So you can actually control expression through changing the promoters around. Okay, so let's answer these questions. What is the relationship between genes and traits?
Is it trait when a gene is expressed?
Okay, so when genes are expressed, they influence the traits. That's right. Genes code for the traits. That's exactly right. Good. So genes tell us what proteins to make, and those proteins control our traits. So we talked a little bit about enzymes. Ribosomes. Oh, not right. RNAs. Sorry. We talked a little bit about RNAs having their own job, and sometimes RNAs can act as enzymes in the sense that they can cut something. All right. So when we call an RNA that is capable of cutting something, we call it a ribozyme. All right. And it basically can cut things. All right. And so these are becoming more and more obvious to us as ways the cell regulates gene expression. So you can imagine if the RNA was made and has been being made into protein, at some point we need to degrade that RNA to slow down the protein production. Well, these ribozymes will recognize the RNA, bind it, and then cut it up to pieces, which will then be transferred to the lysosome to be further broken down into their building blocks. So this is RNA is controlling how much RNA is actually present in the cell at any one given time, so that we're not overproducing proteins that we don't need to be making. Okay, so we've talked about the genetic processing of all of this, and I think we talked a little bit about sickle cell. Did we talk about sickle cell? Yeah or no?
Yes.
Okay, so remember, sickle cell comes from the gene for hemoglobin, right? So normally we make hemoglobin, and they fit into a red blood cell that looks like this. When we have a mutation in the DNA, the hemoglobin doesn't take on the right shape, and it causes the red blood cells to look like this. All right. So let's talk a little bit about mutations and the kinds of mutations there are that then code for these different kinds of changes in the DNA. So the first thing I want to talk about is just the general word mutation. And mutation means there's a change in the DNA sequence. It can come from lots of different sources: UV rays, radiation, smoking, chemicals, asbestos, right? Those kinds of things that all kind of mess with our DNA can cause mutations. And one of the kinds of mutations we're going to talk about are called point mutations. And this is where you just have a change in one base at a time. All right? So we're going to talk about different kinds of point mutations that cause different kinds of outcomes. All right? So one kind is called a substitution. So you have one nucleotide that is substituted for a different one. So there's still the same number of nucleotides, just supposed to be CG, and now it's TA. Or you can have ones that are either insertions or deletions. So now you have more nucleotides, a different number of nucleotides, and you're either inserting or you're deleting nucleotides. All right, so let's talk a little bit about each of these. And we're going to use our hemoglobin gene as our example. So here's our normal hemoglobin DNA, which makes this mRNA, which makes this normal amino acid chain. In hemoglobin, one nucleotide is changed, and that causes one amino acid to be changed. And we already know that that causes the whole protein to fold wrong and the cell to be wrong. So there's different kinds of point mutations that are substitutions. So we're going to talk about 3 different kinds: silent, missense, and nonsense. And do you have to know the difference between them? You absolutely do. Okay? So silent mutations, let's see. Well, we'll read this together and then we'll look at the example. Silent mutations mean that there is a change in the DNA, but because of the redundancy of the genetic code, there's no change in the amino acid sequence. All right? Whereas a missense mutation is a change in the DNA that results in a change in what amino acid gets put into the amino acid sequence. So you can see here we changed the DNA, but no difference in amino acids. Here we changed the DNA, and we did get a difference. A nonsense mutation, so don't get missense and nonsense mixed up. A nonsense mutation means we change the DNA, one nucleotide, but that one nucleotide change results in a stop codon. So there's a mature ending of the protein. All right, so silent means it makes the same amino acids. So even though there's a change, it's still a mutation because there's a change. It's silent because we don't see it. Nonsense means we actually make a stop, and missense means we change the amino acid. Now, is that always bad? Well, here you have 2 different kinds, conservative and non-conservative. It depends, right? If you were changing from a lysine, which is shown right here, to an arginine, heck, they look pretty much the same, don't they? So the cell might not really care and fold it the same way. The protein might fold in the same way. However, if you change from lysine to threonine, this looks pretty different. And so the protein may not fold the same way. All right, so I want you to be able to distinguish one from the other. So if we have a change in the DNA that doesn't change the amino acid, that is a silent mutation. It's still a mutation because there's still a change in the DNA. If we have a mutation that does change the amino acid, so notice we were met-lys-phe-gly, now we're met-lys-phe-ser, that's a missense. We're getting a different amino acid. It may fold the same, it may not. We don't know how it will function yet because it depends on what amino acid it changes to. This is what happened with sickle cell. It was a missense mutation that did cause it to fold a little funny. And then we have a nonsense mutation. So instead of met-lys-phe-gly, we have met-stop. Well, that's obviously going to be a non-functional protein altogether. Everybody see the difference between the 3? Any questions? All right, now insertions and deletions are way more tricky because now we're going to be either adding or losing a nucleotide. And the reason why these ones are tricky is because we're going to basically alter what's called the reading frame. So I'm going to pull up another picture I can. if this was my normal sequence and these were my, amino acids or mRNAs or whatever, but it read this way, the one big fly had one red eye, right? A missense mutation would just change it like this E to a Q. So it's like one big fly had one red eye. You probably would maybe figure it out, maybe not. A nonsense mutation would stop it early. So one big and then nothing else, right? A frame shift mutation is going to add a letter in such a way that it's going to shift all the letters down one, right? So now everything after that insertion doesn't make sense anymore. So instead of saying the one big fly had one red eye, it's going to say the one down air day, right? It doesn't make any sense anymore. It's still making protein, but not the right proteins and the right amino acids in the right order. Or as if you had a deletion, right? You could have a deletion of 3 nucleotides, or you could have a deletion of 2. Here's an example. Here's where you have an insertion of just 1 nucleotide that causes me to have an early stop codon. So frame shift mutations can result in nonsense mutations as well. Here's where I delete 1. And so notice if I delete right here, I'm going to change every single amino acid after that. Maybe never even get to a stop codon because now I've ruined that. All right. However, if I delete or I insert in sets of 3 nucleotides, I may not have as big of an effect than if I do it 1 or 2. So insertions and deletions of 1 or 2 nucleotides are far more dangerous than an insertion of 3. Because I'm missing the lysine here, but I still have everything else in the right order, and who knows, maybe it'll still fold correctly. Okay? So those are our kinds of mutations that I want you to be able to tell the difference between. And like I said, things that cause these